So we prepared some questions.
Okay.
Okay, so we know that your career has been developed around research in the field of DNA replication and cell division. Do you still work around these subjects?
So the paper is about how DNA replicates when it encounters some kind of a problem. So with the replication, they predicted it, and then Arthur Kornberg discovered DNA polymerase, which was the central enzyme. But when it turns out that DNA is replicated by a protein machine, and that was what we really did, published that. But then we realized that getting DNA to replicate accurately, this is really a challenge for the cell because there are all kinds of chemical accidents that happen. And one of the kind of accidents is like the chain may break, and what are you going to do? Mm-hmm. So you hook it up to genetic recombination mechanisms. The cells mastered all this stuff. It's amazing. And so we had reconstructed some of that process in a test tube with purified proteins, and that's what we published on. The cell manages to make only like one error in a billion replication events, right? Isn't it amazing? I can't do anything. I make one mistake every 100 times. So the cell makes like one error in what the nucleotide sequence is that after DNA replication, every billion base pairs, and without that kind of fidelity, you couldn't have a large organism like ourselves. And I think that's described in our textbook. So as larger and larger organisms evolve, more and more mechanisms had to evolve that would fix any kind of problem that happened during DNA replication. And when we started this, 1998, I started working on these problems. We had no idea that any of this. We thought it was all fairly simple, but the amazing aspect of the fidelity of DNA replication, given all the constraints, the problems that exist out there in the world because of simple chemistry and the kind of accidents that will happen inside of a cell. It's really incredible that life has been able to say who could talk and walk and think. I mean, this is so the wonderful part about studying molecular biology is that the mechanisms are so impressive, and we don't really understand so much. So that young people still have a lot to do.
One of the highlights of your career was your published book, Molecular Biology of the Cell. At what point did you realize that you wanted to write a book?
So it wasn't me. I was recruited by Jim Watson. It was Jim Watson's project, and he had the idea, and I didn't really want to do it. I had no idea that I wanted to do anything like this. But he made it very appealing because he said that, well, I've got it all organized. It's always only going to take 2 months. 1 month the first summer, 1 month to the next summer, and we could do it. We were starting in my house in Martha's Vineyard on Cape Cod, and I knew some of the other authors. Martin Raff was a friend. So, you interact with your friends and you have a good time and all this stuff. And it's going to be easy. The main thing was going to be easy. So I said yes, I would come. We started in the summer of 1978. And we spent the whole summer, I guess it was 2 months really, preparing 2 chapters. Each Martin Raff and I each prepared 1 chapter. I worked on the biochemistry chapter, and he worked on the immunology chapter. He was an immunologist. So
Mm.
I didn't know any immunology, and he didn't know any biochemistry. He's an MD. So what was really important in our book is that, first one author writes something, and then another author who doesn't really know anything about it tries to understand it, and and then eventually edit it so that that author could understand. Then goes a third author. So most textbooks had been written before that time by people. There'd be a big textbook of whatever biochemistry, but different chapters were written by different people in different styles. In the 20th century, it changed to a lot of electron microscopy was added to that. So light and electron microscopy then were the basis for cell biology textbooks that existed, a few of them, which were basically just descriptions of what people had learned from the microscope, and didn't really try very hard to connect that to biochemistry or molecular biology. So, I mean, the beauty of the Watson and Crick discovery, which inspired my generation, in 1953 discovered the DNA double helix, was connecting basic molecular knowledge and chemistry to, in that case, what the chromosome does. The people in cell biology had figured out what this chromosome does, but they figured it contained DNA, but they didn't know how it was possible that the chromosome could carry the information to make a cell. So, or that how it could carry information between generations. I mean, it was all complete magic, and the Watson Crick paper was like a bombshell because people had struggled with thinking about this for many decades, and it just completely puzzled and stymied. But all of a sudden, now you could see your way forward to a molecular explanation of what we knew about how cells work. And so Jim Watson's, so he was the major force in driving that discovery. And by 1976, or 77, when I got the telephone call from him asking me to do this book, he had correctly inferred that now is the time to try to do for other parts of the cell, like the endoplasmic reticulum and the lysosome, and try to do the same kind of thing that they had done for chromosomes, make a molecular explanation. Our research labs were affected by all the new things we learned. So I had been working on the molecular chemistry of DNA replication using a bacterial virus as a model. And that because of all the stuff I had been doing in the textbook, we started working on the structure of the cytoplasm in the fruit fly embryo, because somebody in my lab was interested in this. But all of a sudden, if I hadn't written the textbook, I would have never created the second major area of my lab. So from 1978 or 1980 on, we started this new area of trying to work on how the cytoskeleton organizes in times all the event in the early Drosophila embryo, which is a huge cell with many different nucleuses many different nuclei in the same cytoplasm. If you watch it, it seems amazing. So, I would never have tried to figure that out without having worked on the cell biology textbook, which made me recognize what an important problem this was, and gave me enough knowledge to feel that I could hope to make a progress on that issue. So I think it's true of all of us. All of us who worked on the textbook had good effects on our science.
I think the last thing we wanted to know is what would be your advice for future scientists that are starting their careers now?
really critical thing about being a scientist is discovering what's an important problem to work on. And it's often not the problem that you worked on before, because that's already been worked on. It's the most important problem to work on is something we really don't understand. It's really critical for moving forward with our understanding of life. And to get a feeling for that requires some sophistication and feeling about biology. What's really important, what's not. Anyway, it's been great talking to you. You've been very impressive group of young students. Just like I'm very impressed with Brazilian students.
Really?
Yeah.
Thank you. And we would like to thank you, Mr. Alberts, a lot for this interview. We were so pleased to talk to you.
Oh,
And,
you're welcome.
thanks a lot for this.
Thank you.